todos. Es un honor darles la bienvenida al First International Seminar 2026 del programa de doctorado en Ciencias de la Salud de la Facultad de Medicina de San Fernando de la Universidad Nacional Mayor de San Marcos. Hoy reunimos a investigadores, docentes y estudiantes de pre- y posgrado con un propósito común: fortalecer el diálogo científico internacional y promover la investigación en salud global. Vivimos en un mundo en rápida transformación donde el cambio climático, la urbanización y la movilidad humana están modificando los patrones de enfermedades infecciosas. En este contexto, las enfermedades transmitidas por vectores representan uno de los mayores desafíos emergentes para la salud pública. Es un verdadero honor presentar a nuestro conferencista internacional invitado. Nos acompaña el profesor Matthew Baylis, PhD, profesor de Epidemiología y Biología de Vectores de la Universidad de Liverpool. Su investigación ha contribuido significativamente a comprender cómo los cambios ambientales influyen en la dinámica de transmisión de enfermedades emergentes. Professor Baylis, thank you so much for being here at the University of San Marcos. And now, without further ado, please join me in welcoming the Professor Matthew Baylis. The floor is yours.
Thank you, Yvonne, and thank you everybody for coming to hear me today, and thank you to people who've organised my visit. As you heard, I'm a professor from the University of Liverpool, recently retired, but still carrying on some work. It's my pleasure to give you a talk today about how insect-borne or vector-borne diseases are changing rapidly. And this is very much from a UK or European perspective, not so much a South American one. I will try to speak slowly for obvious reasons. If there's something I say you don't understand, please interrupt and I can try to explain. So I'm going to talk about vector-borne diseases, but first of all, I want to make clear that there have been enormous successes over the last few years and decades in vector-borne diseases. So, for example, the numbers of cases of malaria globally are much less than they were in the past. Sleeping sickness, which is spread by these tsetse flies. This is what I started studying when I first entered this field. Sleeping sickness is much reduced now in some parts of Africa. Elephantiasis or lymphatic filariasis reduced, and onchocerciasis, river blindness, again, a big problem in Africa, significantly reduced. So there's some big successes, but in my view, all of these are caused by parasites, and I think that's not coincidence. Where we are seeing changes is mostly in arboviruses or arthropod-borne viruses, and in my view, we are in the age of arboviruses. So even though there's been successes, there's still a very high burden of vector-borne diseases globally. It's over nearly 1 million deaths per year. That's deaths, but of course there's significant sickness or morbidity. And we shouldn't just think of humans. Animals that we rely on for food are also very strongly affected by vector-borne diseases. The one we talk about most nowadays as arboviruses is dengue, with, nearly half of the world's population at risk. And we still have the highest burden, of course, in the tropics and the subtropics. In some cases, the risks are getting worse because of the spread of insecticide resistance. Our vectors are becoming resistant to the insecticides we use to treat them. But what we are seeing a lot now is vector-borne disease risks increasing in places where we didn't used to see much risk at all. So temperate regions such as Europe and the UK are now exposed much more to vector-borne diseases. These are just three of the types of vectors that I've worked on in my life. Ticks a little bit, mosquitoes more, but in particular, Culicoides midges is my speciality, and I'll talk more about those later. So from a British perspective, I put this slide up because most people in the UK would say that we have never had malaria. We think of malaria as a disease of the tropics, but that's not true. We used to have malaria in the UK. These are the writings of some of our old British writers, a man called Chaucer, and he used this term ague, which is another word for malaria, and tertian fever. And some forms of malaria come back every 3 days, and that's tertian fever. And so he was writing about this in the 1300s. And then our most famous writer of all, Shakespeare, in one of his plays, he mentions ague as well. So this would be a surprise to British people. How did these old writers know about malaria when it's a tropical disease? Well, the answer is it wasn't a tropical disease. We used to have it in the UK, but we got rid of it. So interestingly, and most British people would be very surprised, our last outbreak of malaria in the UK was just over 100 years ago. And we still have places in Britain with lots of anopheles, the mosquitoes that transmit malaria. Our last case was within living memory. The last locally transmitted case was some 70, 80 years ago. So we had malaria, we got rid of it, but not that long ago. But we've completely forgotten about it. And now we think of malaria as a disease just of the tropics. Of course, we still get cases coming in with travelers. So we get a lot of malaria in people coming from holidays or visits overseas. But also we do get some transmission to people living near airports. If you live in London, near our big airports, planes coming in from other countries can bring mosquitoes that cause malaria. So you do get some cases. But as I said, we mostly got rid of it. And we got rid of it through changing our landscape, through draining our wetlands and improving our housing so that mosquitoes don't come in. But maybe a big change is that, for example, our baños, toilets and so on, used to be outside. My grandparents, they would have gone outside the house to go to the toilet day or night. But now, of course, our sanitation is inside the house. So now people are not going outside in the night. So these sort of changes drove malaria away in the UK, as well as some other things. So that's malaria. We've also had just one previous outbreak of an arbovirus, which was yellow fever. And this was from a boat coming in from Cuba in the 1800s. And basically, there was an epidemic of yellow fever on the boat. And it brought Aedes aegypti mosquitoes into a town called Swansea, which is in Wales. And those mosquitoes then spread around the town and caused an epidemic of yellow fever in the UK. But that's it. For arboviruses, this is the only outbreak that we've ever had. One in the 1800s, and that was caused by someone bringing mosquitoes in. By a boat bringing mosquitoes in. But now things are changing fast. So here's just a few slides on what's happening in Europe with various mosquito species. The one that is a big problem globally is Aedes aegypti. That's not such a problem in Europe, a little bit around the Black Sea, and it's on an island here, Madeira. Otherwise, fortunately, still limited. But other species of Aedes are invading into Central Europe. But the most interesting one is this Aedes albopictus. So I don't know if you have this in Peru. You may do. I'm getting a nod. You do. So this is what's happened with Aedes albopictus. So this is the Asian tiger mosquito. So it's from Asia. It invaded Europe first in the 1970s into Albania, which is here, and then in the 80s into Italy, somewhere up here, no, that side. 2 invasions. But you can see that just 15 years ago, it was still very limited into this part of southern Europe. And then 13 years later, Aedes albopictus is spread over a huge area of Europe. And red is where it's present all the time. Yellow is where it's sometimes found. And you can see that it's just been found in the UK. So this is, one of the great vectors of dengue, chikungunya, even yellow fever, and other pathogens. And now we are beginning to see it very close to London, but it's a big risk over much of Europe. That's in 13 years. What will it be like in another 10 years? It's changing very fast. And it's not just the mosquito. So the vector was found in Paris a few years ago, but then just 2 or 3 years ago, they had the transmission of dengue in Paris. And I do find this remarkable, that you could go to Paris for the weekend now and come back with dengue. And that's unthinkable, for the vast majority of my lifetime. And you can see there's cases of dengue over small numbers of cases over other European countries as well. So this is changing in Europe. Dengue is becoming established in parts of Europe and perhaps one day in the UK. Another virus of interest is West Nile virus. This is one famously that invaded the United States in 1999. But it's been in Europe for much longer. It was first discovered in Europe in the 1950s. And it had occurred in a few places in southern Europe for a long time. And we used to get some cases of West Nile. But you can see, again, spread of West Nile in recent years over larger areas of Europe. And importantly, including much further north than previously. So here now, cases of West Nile in Germany. And that, of course, is of the same latitude as the UK. So this was alarming for us. And it made clear that we might be at risk of West Nile in the UK. This is transmitted by different mosquitoes from the ones that transmit dengue. These are Culex mosquitoes. So we were aware that there must be a risk from West Nile in the UK. The same mosquitoes transmit a virus called Yasutu virus. This is a bit milder than West Nile, but similar in other ways. So this was found in London, I think you can see here in 2020, so 5 years ago, 6 years ago. This was the first example since yellow fever in the 1800s that a mosquito-borne arbovirus was found in the UK, being transmitted in the UK, in the heart of London. This is in Regent's Park, in the absolute centre of London. Fortunately, it's a mild disease, but we've had no cases in humans, but it is killing our birds, these blackbirds in particular. And then after Yasutu had been discovered, maybe last year, it was announced that West Nile was also now present in the UK and had been found in mosquitoes in a part of the UK. So we have the dengue vector invading the UK and cross over much of Europe. We have Yasutu virus established in the UK, and we have West Nile virus now appearing to appear in the UK. So it's not just mosquitoes. There's also ticks. I just heard someone say earlier they didn't know we had ticks in the UK. We have lots of ticks in the UK. And we have one called Ixodes that transmits borreliosis, Lyme disease, which has become a big problem in the UK. But a student of mine, I'm proud of this because this is perhaps my only real discovery. A student of mine used a One Health approach, and we discovered this other disease transmitted by ticks to be present in the UK. Tick-borne encephalitis virus is a flavivirus, like dengue virus, like yellow fever virus. And we did not believe we had this in the UK, but we knew it was a big problem in parts of Europe. This student did a very nice One Health study where she got hunters of deer, those animals and deer, to supply blood. And she tested that blood and she found evidence of flavivirus antibodies. And then she went to follow up where the deer had flavivirus positive samples, blood samples. And through that, in the end, we found ticks with this tick-borne encephalitis virus, and so discovered that actually this virus was now present in the UK. We've only had a very small number of cases in humans so far, so it's not such a public health issue. And then looking more widely across Europe, lumpy skin disease is a viral disease spread by various insects, culicoides, biting flies. This was exotic to Europe, but in the last couple of years, it's invaded Italy and then France. It causes economical problems through the effects you can see on an animal here. So this is an invasion of a new disease into Europe, a new vector-borne disease into Europe. Another one, epizootic hemorrhagic disease. This is another culicoides midge-borne disease, to ones I'm going to talk about later. This was present in the United States, and it was found in Israel as well, but this has also spread into parts of Europe, Italy, and then a large outbreak in France in the last couple of years. And then thinking more globally, another good example here is this terrible disease, Japanese encephalitis virus, another flavivirus spread by mosquitoes in a cycle with pigs and birds, and then causing a nasty disease in people. This was over much of Southeast Asia, but not Australia, other than some cases in the north. But just a few years ago, it invaded Australia. So we're seeing vector-borne diseases moving in the northern hemisphere and the southern hemisphere. And then the final example I'll give you, out of many others, is Oropouche. Oropouche is the reason I'm here, because I'm working with Yvonne Ramirez here. But Oropouche virus is a culicoides-borne virus discovered in Trinidad in the 50s. Here I am with one of my old students in Trinidad last year trying to catch the vectors, which we didn't manage to do. But as you can see, after being discovered in the Caribbean, Oropouche is known to be present over the Amazonia region. And it's transmitted most famously by a species of culicoides midge, Culicoides paraensis. This is the only known human disease, viral disease, transmitted by midges. Midges are big vectors of animal disease. This is the only significant human disease transmitted by midges for various reasons. So this disease has been in the Amazon for 70 years, but a couple of years ago, there was a large outbreak in South America, including Peru, but also in Brazil, and it spread to Cuba. And that's caused a lot of alarm because it turned out that Culicoides paraensis had managed to spread to Cuba and was then transmitting the virus there. So we had lots of concern in Europe. So when Yvonne contacted me about a possible study, I said I was interested in oral pooch, although I didn't know anything really about it. So it was a pleasure to become involved. So I'm here now to visit Yvonne, and a couple of days ago, I went out into the field with Yvonne, and I realized that my function is twofold. Firstly, I'm a mentor and supervisor. And secondly, I'm bait. I turned out to be the most attractive person for the midges. So here I am being hoovered, and I can tell you now that my legs and my arms are covered in hundreds of bites, and it's still painful. But that's an example of what we caught. It was amazing. In a few minutes of this sort of hoovering, we catch whole tubfuls of midges. That was a great experience. So that's a bit of background to show you just some examples of how the vector-borne diseases seem to be changing. I can't talk about all the examples, but that's just some. My main research has been about, why is this happening? What drives it? And how can we start to predict what's going to happen in the future? And for that, we need modeling. So I guess my main expertise is in, one of my expertises is in modeling vector-borne diseases, and in particular looking at how we can model the effects of climate change. So an important question is, how do we measure risk from vector-borne diseases? If we're going to model it, we need to be able to break it down into its pieces. So there's really three things that are important for measuring the risk of vector-borne diseases, and I'll try to explain them. Firstly, vector competence. This is simply how able are you to infect some vectors with a certain pathogen. If you feed Aedes aegypti mosquitoes with blood with dengue virus, do you infect 100% of them? Do you infect 50% of them? Do you infect 5% of them? There's genetic aspects which will determine how susceptible the mosquitoes are, or the midges are. And this varies from species to species and from virus to virus. And that's an important component. And that explains, of course, why tsetse flies don't transmit malaria, and mosquitoes don't transmit sleeping sickness, because different levels of competence. That's one. Second most important thing is, of course, distribution and abundance. Do you have the vectors, and how abundant are they? How many are they, especially relative to hosts? But the third thing is, how able is your vector to transmit a pathogen, or a population of vectors to transmit a pathogen? And we call that vectorial capacity, and that's different from vector competence. Vectorial capacity is a different thing. Importantly, from the modeling perspective, so these two here depend on climate. Warmer climates, usually more abundant vectors and so forth. So we can do research to quantify that. But also climate affects how able a vector is to transmit a pathogen. And I'll explain that in more detail. So to understand that, you need to understand how vectors become infected. Just very briefly, you'll all be aware that when a mosquito here feeds, obviously it sticks its mouth parts in, and then it sucks up blood into its crop or its gut here. It's important to understand that just before it feeds, of course, it injects you with saliva to anesthetize you, to help blood flow. And that's the point at which, if the vector is infected, it will be inoculating you with a virus or malaria or whatever. Otherwise, of course, things are flowing the wrong way. So it's at that very start that it might infect you as it's injecting you with saliva. But once it feeds, it takes the blood up, maybe you're infected, and now it's taken your blood with some dengue virus or a malaria parasite into its crop. So that pathogen has got to travel from the crop through the cell wall into the body, and it's got to travel through the body, and it's got to then get to the salivary glands, and it's got to then cross the wall and into the salivary glands. So this takes some time. You know, when you're bitten by a mosquito with dengue, it goes straight into your bloodstream, and you will get dengue in a relatively short time, maybe 4 to 7 days. But the other way around, when your blood infects the mosquito, it takes much longer because it has to go on this journey through different parts of the body. And that can take 7, 14, 21 days, depending on the temperature. That time we call the extrinsic incubation period. How long it takes from this vector feeding to it becoming infectious, the EIP. So this is quite a complicated slide, but what I want to show here is, imagine, so what we're seeing here with these shapes is a vector emerges, and it takes a feed here, and then it runs through that EIP. This is the time that the pathogen is spreading through its body, and it's going to take a certain amount of time. So it's at this point that this first vector has become infectious. And you can see, as I've drawn it, that in the rest of its life, it takes 4 feeds before it dies. So this means that this vector has 4 opportunities to transmit its infection to another person or an animal or whatever. Now, if we change some things, we can change that risk. So if these vectors feed more frequently, maybe every 3 days rather than every 4 days, or something like that, everything else is the same. It's feeding more frequently. Now it's got five opportunities to transmit before it dies. Or if it takes less time for the virus or the pathogen to get to the salivary glands, so a shorter EIP, everything else the same. Now we've got 5 opportunities to transmit before it dies, or a lower mortality rate. If the vectors simply live longer, then everything else being the same, again, we've got 5 opportunities. So what I'm showing here is that these 3 things, how often vectors feed, the EIP, and the mortality rate, determine the risk that a population of vectors will transmit an infection, because it determines how many opportunities they have to feed after they become infectious. So when we do lab studies or field studies on those things, we find that they're all affected by temperature. The biting rate, the mortality rate, and the EIP are all sensitive to temperature. And this is why then changes to temperature have a big effect on the risk of transmission of vector-borne diseases by changing these 3 components. But when we put them together, it's not that as they increase, when we put them together, we get this shape. So at low temperature, no transmission because the EIP is just too long. Then as temperature increases, we get more transmission risk, more vectorial capacity, but it reaches a peak. After that, as it gets even warmer, that risk begins to go down. And this is because the mortality is increasing fast as it gets hotter, and so they're not living as long. Even though the EIP is short, their lifespans are even shorter. So we have an optimum temperature for transmission determined by these three things. But a really important point is that for some insects like tsetse flies, they live 100 days. Ticks live 3 years. For them, it takes very little time for them to become infected relative to how long they live. But if you're a mosquito, or even more, if you're a midge, you may only live for 5 days as an adult, 5 days, 6, 7, 10 days. But the EIP can be 5 days, 7 days, 10 days, 20 days. And so there's a very tight balance between will you live long enough to become infectious? You're a short-lived insect, like a mosquito or a midge or a sandfly. Will you live long enough to become infectious? And that is highly sensitive to temperature, as we're seeing here. And that's, in my view, one of the reasons why we're seeing such changes in arboviruses spread by mosquitoes and midges with climate change. Small changes in temperature having a big effect on the ability of vectors to transmit. So that's the theory. Now I'm just going to talk about my particular expertise in midge-borne diseases, and in particular a disease called blue tongue, which I'm guessing you have in Peru, but it's probably not a big animal health problem. This is an animal disease, not a human disease at all, but it's an African disease of ruminants in Africa. And there's a very similar disease that affects horses called African horse sickness. Very similar virus, transmitted by the same vectors. So these two diseases, actually, blue tongue and African horse sickness, both African, but for 100 years after their discovery, we had 2 outbreaks of each of them in Europe, each one for maybe 1 year. But those outbreaks were exclusively in the hottest part of Europe here, Spain and Portugal, and the hottest part over here in Greek islands. That was it for 100 years. So when I started to work in this area, which was in the 1990s, for me, this was a disease, these were diseases just of hot places. I started to work on African horse sickness in Morocco, where there'd been an epidemic. And it seemed that the risk to the rest of Europe was very small. But everything began to change in 1998. We got another outbreak of blue tongue, but then every year we got more blue tongue coming into Europe. So over 7 years, we had repeated invasions of different types of blue tongue virus. There's 25 different types invading into Southern Europe. And we discovered that the African vector, which had invaded here and here before, had also now spread over other parts of Europe. So a bit like paraensis going into Cuba. The vector spreads, the vector-borne disease spreads. And we saw the same. The vector spreads into Italy and some parts of France, and the disease spreads. But interestingly, there was also blue tongue in this area over here that we call the Balkans, or Bulgaria, where this African vector was not found. And that should have alarmed us more than it did. Because then in 2006, we suddenly got blue tongue in Northern Europe for the first time ever. And then in 2007, it spread over a much larger area, including into the UK. 2008, it was all over Western Europe. So we had a huge invasion of blue tongue over 10 years. So I started to work on these diseases, I said, in 1993. At that time, There'd never been a case of any midge-borne disease in Europe north of Madrid. And 15 years later, we were getting disease in Scandinavia, in Sweden. There's an amazing move north. And the diseases are now endemic in Europe. This African disease that was exotic for most of my research career is now endemic in Europe. So a big change there. So a lot of my work, some of my work was, can we explain this? Okay, we thought maybe it's climate change, but we didn't know whether it's climate change. And it's very hard to prove, but we did do some research in this area. So I said that I'm a modeler. So we did a model for the risk of blue tongue. This was modeling that vectorial capacity. And I mentioned before, and the reproductive number. So without going into details, what you're seeing here is that line is the long-term average risk in our model. And depending on the climate, a year is either below the long-term average in blue, or it's above the long-term average in red. So let's start down with Southwestern Europe or Southern Europe. So here there'd been some history of culicoides-borne disease, blue tongue and African horse sickness, I mentioned. Those are the stars. And you can see that going back maybe 50 years, okay, maybe there's some evidence of change with more red years in the later times. But importantly, from my perspective, where there were outbreaks of disease, so the stars, that tended to be where we had the red bars. So that reassured me that actually our modeling is working quite well. And our climate-driven model explains, to some extent, when we get outbreaks of disease. But then look what happened in Northern Europe. So a long period where it's all much the same, and then around the mid-1990s, we just got a surge in that risk of the transmission of blue tongue, measured by our model, entirely driven by climate. And you can see that in the last year of our modeling, that was the year of the highest risk, and that was the year that we got blue tongue into Northern Europe. So for me, this was fairly convincing evidence that climate is playing a role in when we get outbreaks of disease in Europe, and that climate change we're seeing here is real and increasing that risk to Europe. So that's one approach we've taken. And then another approach is a much more complicated model where we wanted to look at the spread of the vector from farm to farm, because then we can begin to ask much more important questions. This is some work that Yvonne is following up in one way, with some more approach. I won't go into any details here, but we spent many years building a very complex model that has every farm in the UK, some 100,000 farms of sheep and cattle. And we, in our model, so this shows how animals move, where animals move from one farm to another farm in the UK. You can see that it's sort of localized. Animals tend to move within their region, but sometimes there's long distance movements. Those long distance movements can take a virus right across the country. So in our model, we needed to look at two ways that the virus can move. One is just in the vectors, spreading from one farm to another farm. And the other is by the movements of animals. Maybe this farm here has got infected, and then the farmer moves his or her cattle to a farm up here and could take the disease with them. And we're lucky in the UK because ever since we had a big outbreak of another disease, foot and mouth, 25 years ago, we have recorded all of the movements of all of our animals. Every cow that moves from one place to another, it's recorded, and groups of sheep. So we have those data. So we're able to use those data to look at how our animals move around and how they might carry disease. And then in our model, we can do lots of other things. like impose control measures and look at different years. So here's some simulations now of the future, the past and the future. So what we're doing here is we're, in our model, dropping a virus into one farm and then allowing, under the climates that we're told we will get at different times, looking at spread. Now here we have animal movement restrictions on. So this is just the movement by vectors from farm to farm. But you can see that in our model, with different, as we go forward through the century, with different temperatures, we get slightly bigger outbreaks, where we get bigger outbreaks. So what we then did here was to run thousands of simulations for every year with different climate change models and different scenarios for how much CO2 release there will be. So there's a lot of uncertainty, but this was ultimately what we found. So what we have here is the past, but then from here we were predicting into the future, and we have all those uncertainties. So we have these sort of clouds, two scenarios: higher CO2 release, less CO2 release. But what you can see is that under both scenarios, we get increasing scale of outbreaks as we move forward through the century in the UK. And perhaps importantly, this little dot here, this was 2006 when that blue tongue appeared in Northern Europe. It was an exceptional year. So exceptional, it triggered a huge epidemic. That was exceptional in 2006, but according to our model, under this scenario, the high CO2 scenario, it becomes normal by about 2070. It's what we would expect, every year. So concluding there, what we see really is a significant increasing risk of blue tongue in the UK as we go forward, driven by climate change. But is it a problem? One of the advantages of our model is we could test different control options. The main control option we use at the moment is you can't kill midges on farms. There's too many. There's no insecticides that will do the job effectively. So all we can really do is stop animals from moving from farm to farm and try to contain an outbreak. And that's what we're showing here, is that under current climates, this is when we allow animals to move. But if we put on animal movement controls, we get a much smaller outbreak. And even in the future, animal movement controls are still effective at reducing the scale. Still bigger, but animal movement controls remain effective. So the point of this work was to say, look, in the future, we think the risk of blue tongue and the scale of outbreaks will increase, but we don't need to panic, because what we're doing now should still be pretty effective in the future. That's not the case with all vector-borne diseases, of course. Okay, I'm going to finish just with one other area. I've talked about blue tongue invading Europe, but one of the key questions is, where on earth is it coming from? My research and other people's research suggests that through climate change, it's become easier for blue tongue virus to stay in Europe or to stay in the UK, to persist. But how did it get there? We have these repeated invasions. Where did it come from in 2006? And it's not just, so that one in 2006 is blue tongue virus 8. It had not been seen by anyone anywhere in the wild for 50 years, since the 1960s. Suddenly it appeared in Northern Europe. But there's other blue tongues which now keep appearing. So we've also had blue tongue virus 3 and blue tongue virus 12 just in the last few years. Both of them in the Netherlands, and both of them also near to Schiphol which is the big airport in Amsterdam. And there's another disease that I haven't talked about called Schmallenberg, transmitted by midges. This was a brand new disease, newly evolved in 2011, which was first found in a village called Schmallenberg in Germany, but also quite close to the Netherlands border. And so it's intriguing to wonder why the Netherlands so many times. Is it coincidence or is there something going on? Well, what are the possible routes of these viruses? They could come in on live animals. We know live animals come into airports. Could there be a midge on a plane? A bit like we know mosquitoes can board planes. Midges aren't really attracted. Most midges that transmit blue tongue are not feeding on people, so probably not. They can get blown on the wind because they're very small. Or maybe there's other things. One intriguing area that I've done some work on is this one. So this is a map of the global movements of flowers, cut flowers. So Colombia, your neighbor, is a huge supplier of cut flowers to the United States. Maybe Peru does this as well. Does Peru grow cut flowers for export? Like roses, big industry. And in Africa, Kenya and Ethiopia are huge suppliers of cut flowers to Europe. But nearly all of the cut flowers from Africa come into Schiphol Airport in the Netherlands before they get exported to other parts of Europe. And we did some research on this because we know that, so 60% of all flowers entering the European Union come into the Netherlands first. And look at that volume, 273,000 tons of flowers in 1 year. So that shows you that volume. And we know that many of these consignments have insect pests. So we did some work at a Kenyan flower farm. This was very complicated because the flower farms don't want to be identified as a source of diseases, perhaps. But this one did allow us to do some research there. And sure enough, we found that there were Culicoides vectors of blue tongue virus around the farm. We did catch some inside the greenhouses where the flowers are. And then what happens with the flowers, of course, is that they're cut and they're taken into a plant and they're wrapped up in cellophane or plastic and they're chilled and they're driven straight to the airport. 10 hours later, they're in Schiphol. It's a perfect way to carry a vector around the world. And although we didn't find any in the chilled areas, the cold areas, it does seem to me that this is probably one of the ways that we're carrying vectors around the world in the cut flower trade. So I'll finish with a brief summary. So I started by saying, we've made great progress with some vector-borne diseases, but various aspects of globalization are increasing the risk of other vector-borne diseases. We're carrying vectors to new places. We're carrying diseases to new places with our global movements and our global trade. And climate change is playing a role in enabling these vectors to establish and diseases to be transmitted better by those vectors in new places. It's my belief that arboviruses are particularly sensitive to these changes, and that's why because of their short lifespan of those vectors relative to the EIP, and that's why we're seeing so many arboviruses emerge, because new ones will arise, even though we might think we can handle things like blue tongue virus. Schmallenberg was a completely new virus that arose in 2011, transmitted by midges, and swept across Europe in weeks. Maybe the next one will be much more significant, like COVID. And so we need new solutions, technological solutions to deal with these things, better vaccines, new vaccines, drugs, insecticides. And something that I saw this morning at the NIH lab, that use of new technologies like Wolbachia or gene drive so that we can get on top. We're going to be having more risk and more transmission of vector-borne diseases in the future, and we're going to need new approaches to control them if we possibly can. Okay, I will thank you for listening to me. I hope you understood what I was saying, and I hope you found it interesting.
Muchas gracias, profesor Baylis, por esta excelente presentación. Abrimos ahora el espacio de preguntas y comentarios. Ok, por favor, levanten la mano. Ya. ¿Cómo?
Gracias.
Thank you. Excellent presentations. I'm very worried. You have explained that these vector-borne diseases, which were never being in the past in Europe, now they are there and they are spreading. Rinderpest has been the only disease in animals which has been eradicated from the world. Is there any chance that any of these vector-borne diseases can be eradicated?
Yes, I mean, there are some that could be eradicated. I mean, I think some of the parasitic ones like sleeping sickness, lymphatic filariasis, some of those ones which are being successfully controlled. I think that's where there's hope. I think these arboviral diseases are much less likely to be eradicated. What makes the eradication of a disease possible is not many hosts, not many animal hosts or human hosts, because if there's a wildlife reservoir, it's just impossible. If it's smallpox, just with humans, managed with humans. If it's rinderpest, cattle and buffalo, maybe some other wildlife, but nevertheless, it's a limited number. You can do it. With many of these other diseases, where they infect birds, other mammals, then it's clearly impossible. Something like dengue, okay, that's mostly human, but we know it can also be in monkeys. It could be much harder to try to eradicate. So I do think it's going to be very hard. And the other important aspect to eradication is how important is that disease perceived. When you've successfully controlled the disease, there's now very little of it. And it costs a lot of money to go and eradicate the last few cases. And that money people might think that money is better spent somewhere else. So the political aspect is also very hard to really eradicate. Rinderpest was eradicated because it's so devastating to animals. Smallpox, clearly, also devastating in humans. So for those, the political argument was clear. But I think with some of these other diseases, once you've controlled it, politicians would decide to spend their money on something else rather than spending all their money on actual eradication. So for different reasons, I think it's possible, like sleeping sickness, that's an example where some types of sleeping sickness could be eradicated in the future. The one that affects only humans, and because we know it's so devastating in African populations, that could be, and the technologies are there now to kill the tsetse fly vectors. And then with treatments for humans, perhaps with enough effort, that one could be eradicated. Similar to polio. Also, when the politicians decide they'll do it, you can perhaps see that end, but it's very challenging.
And another question. You said that there are very few cases where people around airports are getting infected because this, let's say, malaria.
Mm-hmm.
I used to travel a lot from Europe to malaria countries, At least in the last 2 years, I haven't been there, but every time I returned to Europe, the staff had spray in the cabin, these insecticides, which annoy everyone. But do you think that spray can kill all these mosquitoes because they are coming to Europe?
Yeah. So we don't do a lot of spraying, and I think there should be more. I think it's a very sensible thing to do. You know, one of the things that frustrates me about the UK is that we're an island, and we could be like Australia and have very strict biosecurity to protect us ourselves, but we don't. So I do think it's effective spraying in the cabin, I'm sure, to kill mosquitoes that might be on board. But I suspect they're coming in freight as much as anything else, where there's probably not spraying happening. You know, an airplane, there's obviously lots of different parts to it, and there will be mosquitoes in other parts of that plane. So we do have surveillance at our airports for mosquitoes, and we do find them. Even things like Aedes aegypti, we might find in Heathrow Airport sometimes that somehow got into the country. Usually then they clearly don't establish in the UK at the moment, but we have that constant introduction. And I guess that's the worry with climate change, that as our countries become warmer, so eventually establishment becomes more possible. With the other mosquito I mentioned, Aedes albopictus, that has got into the UK, that one seems to be spreading in vehicles. So I said at the start that it was in Southern Europe, mostly in Italy, Albania, Croatia, and so on. But over a decade or so, began to move north, but it moved north up the main motorways. And it seemed to be entering vehicles and establishing at a petrol station, service station, and then moving north like that. And we have now built a tunnel, you know, from France to the UK. And so vehicles can now just come from France straight into the UK. And I suspect that's how we're bringing in that vector. Still too cold. We're still a little bit, it's not settled, not established in the UK, but that will happen with time. So that's a very nice example of how globalization in general and our transportation of ourselves and our goods is carrying vectors from one place to new places.
And Europe, with all the technologies, the economy, money, facilities, the farm control they have, cannot control these diseases. How can you expect that in Asia, in Africa, where they are the poorest countries, can control that?
Well, that's a very good question. You're right. So, you know, these things are very difficult and very expensive to control. Clearly, all countries can do something. You know, there are ways of suppressing vector numbers, not culicoides, but mosquitoes. The technologies are there if you wish to use them. So I guess most countries can do something. But it's an interesting point you make. You know, even highly developed countries like in Europe and also the USA with West Nile, in the end, we're not immune to these problems, and they're not third world problems, they're not developing country problems, they're now global problems, and that's how we should look at them.
¿Alguna pregunta aquí en el auditorio? Entonces pasamos a las preguntas virtuales. ¿Cómo le da, por favor?
Adelante, doctora Lucy.
Thank you so much, Dr. Barron, and thank you so much, Dr. Baylis, for this conference. Definitely the work you are doing on making these projections of the progress of the disease is part of the knowledge that we need to think about and making decisions. No? On dengue, for example, we, in the past years and based on experiences in different parts of the world, we are already in the country using Wolbachia and using vaccination for dengue. No, and we're trying to apply that to control the problem in Peru. Okay, that is a problem that you have, as has mentioned, is the global warming is definitely going to be something present, and it's difficult for us to modify that condition. But what we can do, what can we do? We started to talk about the spraying and so on, but what other measures do you anticipate that we can pay attention to control all these diseases we are having? No, because we're thinking in public health to use all the knowledge that you are advancing, and in that sense, what measures are being tested at this moment that can be used in countries like ours, you know, to control these diseases that are borne, mosquito-borne? Thank you so much again for inspiring us, for the level of commitment that you are having along all these years on these diseases. I really appreciate that.
Thank you very much for your question, Dr. Lopez. Yeah, so that's an interesting question. I mean, what can we do? I mean, I guess, as I see it, you know, we have some big problems ahead through things like climate change and globalization, our movements, which, you know, we are still just accelerating, and climate change is just accelerating. You know, despite everything that we have said for the last 30 years, we released more CO2 last year than any year before, despite everything else. So there's no real sign that that's going to change in 100 years, I think. And also that spread of insecticide resistance. You know, we've had a nice period during the 1900s when our insecticides worked, and we're going into a period where our insecticides don't work very well, and we're not developing new ones anywhere near fast enough. So I do think the net change for much of the world is negative. Just going back to climate change, one important point is that the slide I showed about vectorial capacity means that if you're a cool country, climate change will increase your risk. If you're already a warm country, it may actually decrease your risk because you're over that hump, over that peak. A country like Peru will be interesting because you've got both warm and cold, warm and cooler areas. And so I might anticipate decreases in some areas of Amazonia or the coast maybe, but increases in the Andes, just as we see in the Himalayas, where dengue is now a much more significant problem in Kathmandu than it was where it wasn't in Kathmandu. And now it's a big problem in Kathmandu. So we see similar things in Peru. So that's one aspect of that. There's a lot of negatives. And the positives are, as you mentioned, you know, things like Wolbachia as an exciting new approach to controlling vector-borne diseases. But I do think we need to recognize that the use of Wolbachia is very limited. You know, it's very good with Aedes aegypti and Aedes albopictus. So let's say dengue is something that we might be able to manage better with Wolbachia infected mosquitoes. And you know, there's evidence that it works. But of course, some other vector-borne diseases are transmitted by tens or hundreds of species. Malaria, Japanese encephalitis, West Nile. There's no way that a Wolbachia-based approach could do much at all, because you might spend millions of pounds on one species, but there's all those other species that are also vectors. So I do think those positives around Wolbachia they're there, but they're particularly there for some specific vectors like Aedes aegypti, as I said, and maybe one or two species of Anopheles. So I think actually the future for vector-borne diseases is not great from a human perspective. Things are going mostly in the wrong direction, and that's literally what we're seeing in Europe as we were a temperate climate with very limited risk or exposure to vector-borne diseases in the past, 0 in the UK. And now, I live in a very different world from the one that I grew up in. And I'm afraid, I don't know that we can change that. So that's a bit negative. But, we will manage, I'm sure. I think our future problems are different from these. It's going to be the effects of rising sea levels and other things will be perhaps more, and challenges to agriculture and food supply will be just as important as these risks from vector-borne diseases.
Thank you so much. Thank you so much. If you allow me a comment to your answer, may I?
So, about climate change, there are social determinants that is complicated to affront them. So, what integral strategy will Wackia vaccine fumigator residual upon, and how factible it is, and what is the prognosis for the future? I think it's something related what you already answered, but if you have any further comments related to.
I'm not sure I fully understand, but what I will say is the social aspect of all of this is hugely important. As an entomologist, epidemiologist, I can see maybe solutions that come from my modeling, but in the real world, you have to persuade people, and they don't necessarily want to listen to scientists. Another important aspect is I showed you from my models how we think blue tongue might spread in the future, but that assumes that the only thing that's changing is climate, because climate scientists are able to tell us what they think our climate will look like in the future. But all those other things that change, no one can even begin to tell us. What will our farming be like in 2080 compared to now? So I'm sure the real world in the future will not be the same as the ones in our model. So those other aspects, and in particular social aspects, are very important, but very hard to sometimes get a grip on.
Thank you.
Agradecemos al profesor Matthew Baylis por compartir sus conocimientos y experiencia con nuestra comunidad académica. Este seminario reafirma la importancia de la colaboración internacional para enfrentar los desafíos globales en la salud. Ahora el doctor Eli Barrón, el director del programa de doctorado en Ciencias de la Salud, va a dar las palabras finales.
Okay, just to say thank you very much, Matthew, for this very brilliant dissertation. It's a really hot topic in this moment in climate change, and everything is affecting to us. On behalf of the Faculty of Medicine, I want to give you this recognition for you, Matthew, for this brilliant presentation of the dean. Is as part of the program. You are not just a lecturer with us, you are part of our PhD program here at San Marcos University. Thank you. Thank you very much. Thank you. Muchísimas gracias. Gracias a todos. Nos estamos viendo en otra ocasión similar. Gracias.
Muchas gracias.